Biology techniques. The first thing we'll do is cover a summary of biology techniques that you might see on your biology praxis, and then we'll do a series of practice questions that might be similar to the questions you may see on your praxis exam. Biology techniques summary. Laboratory equipment. New biological discoveries are happening all the time in laboratories around the world. A brief history of biologists and their past discoveries, which form the bedrock for new science, has been presented, along with a discussion of proper data analysis. In order to collect new data from experiments and to add to the discoveries, some important pieces of equipment are used. A few examples will be shown. Measuring, mixing, and heating equipment. Balances are used to measure the mass of objects. A magnetic stirrer is a device widely used in laboratories and consists of a rotating magnet or a stationary electromagnet that creates a rotating magnetic field. The stirrer causes a stir bar that is immersed in a liquid to quickly spin, which mixes the solution. Bunsen burners are used in chemistry to heat preparations in a test tube. They are particularly used in biology to sterilize instruments by passing them into the flame and the air within a radius of 20 centimeters around the flame, essential for microbiology experiments. Hot plates are used for heating lab materials and samples. Sterilization equipment. Autoclaves are used to sterilize laboratory equipment, medical devices, and tools by subjecting them to high heat and pressurized steam that kills bacteria, viruses, and other harmful microorganisms. They can decontaminate some biological wastes. The high pressure raises the boiling point of water, so when it does convert into steam, it will have a higher temperature and more energy to disinfect the equipment. Laboratory ovens are used for sterilizing, dehydrating, and heating samples with exact temperature control and provide a uniform temperature throughout the oven. DNA electrophoresis is used to separate charged molecules of DNA, RNA, or protein based off their size. Molecules move through a gel when an electric current is passed across it. One end of the gel has a positive charge and the other end has a negative charge. The migration of the molecules is from the negative end to the positive end, as the molecules are negatively charged. Smaller molecules migrate faster through the gel than larger ones, leading to the molecules being separated by size. Chromatography is the process of separating components of a mixture. Chroma means color and graph means to write or draw. Two types of chromatography used in biology are paper chromatography, used to separate plant pigments, and column chromatography, used to separate proteins into their constituent amino acids, which have different properties. Paper chromatography. Mixtures of chemicals, sometimes colored pigments, are separated using a partitioning method. The paper in this method is called the stationary phase because it does not move and serves as a substrate or surface for the separation. Substances being analyzed are separated from each other based on a differential affinity to a solvent. The solvent dissolves and carries the substances along the paper. Since the solvent moves through the paper, it is called the mobile phase. Column chromatography is a general term that refers to using a glass or metal cylinder to hold a stationary phase in place, the column, while a complex liquid or gas sample moves through it and gets separated into components. The column matrix is chosen to separate molecules on the basis of size, polarity, or affinity for the matrix, whichever quality is best for the sample mixture. As a sample moves through the column, here's the sample mixture, components of the sample are slowed down to different degrees depending on their physical properties. Individual fractions are removed from the end of the column, drop by drop, and contain molecules that are separated from the mixture based on their characteristics. So in this case, we can drop by drop separate first the red, then the green, then the blue. Both separation methods compare the distance that molecules or substances migrate to known substances. The distance traveled is called the retention factor, or RF. Centrifuges separate molecules having different densities by spinning them in a solution around an axis in a centrifuge rotor at high speed. Centrifugation is used to collect cells, to precipitate DNA, to purify virus particles, and to distinguish subtle differences in the conformation of molecules. Spectrophotometer. Biological solutions and molecules can be studied through the use of a spectrophotometer. Light, ranging from near infrared to visible to ultraviolet, is split into its component wavelengths or colors by a prism or diffraction grating. Individual wavelengths are selected and passed through the sample solution. This is repeated for the other wavelengths of light. After leaving the solution, the intensity of each wavelength is measured. The intensity of the various wavelengths enables the scientist to discover information about biological solutions and molecules. This is particularly useful in studying DNA, RNA, proteins, and enzymes, as they all have different light absorption properties. Microscopy. Many organisms that are key to biology are very small and can only be analyzed through microscopes. Conventional microscopes can resolve structures as small as 200 nanometers. Electron microscopes that use a beam of electrons can resolve structures as small as 0.1 nanometers. Shown below is an electron microscope picture of C. elegans cells, or roundworm cells. Ultraviolet light sources. Common ultraviolet or UV light sources found in a biological lab are transilluminators used to visualize DNA bands in gels, germicidal lamps in biology safety cabinets, and handheld UV lamps. This transilluminator is used to view a gel after completing gel electrophoresis. The transilluminator helps researchers determine the migration of the bands. Lab techniques and safety. A written procedure for proper lab techniques and safety procedures should be present in every lab and be read and understood by all personnel in the lab. Proper safety procedures should include, but not be limited to, proper supervision at all times is required. All personnel injuries or illnesses should be reported immediately to the instructor. Personal apparel should be appropriate and protect personnel from potential dangers. Dangling jewelry and headphones are prohibited. Lab equipment should be well maintained and checked frequently. Electrical safety is paramount. Safety equipment such as fume hoods, fire extinguishers and blankets, eye wash and shower stations, and first aid kits should be present as required. The lab procedures should be discussed ahead of time so all students know their role. Lab equipment should be taken from its safe storage spot and set up in accordance with the manufacturer's instructions. Only plug in equipment with the permission of the instructor after it's been inspected. The instructor should demonstrate the procedure before the students attempt the experiment. Any deviation from the procedure must be approved by the instructor before attempting. All equipment should be returned to their storage after the completion of the experiment. Handling of acid-base solutions and caustic materials. Chemicals can potentially pose a large safety hazard. They must be handled correctly to minimize the hazards. Never leave the caps of chemical bottles open or lying around. Flammable chemicals should be stored in dedicated cabinets. When diluting acids or bases, always add the acid or base to water during preparation to prevent splattering or boiling. Store volatile chemicals in a ventilated cabinet. Acids should be stored in a dedicated acid cabinet, and nitric acid should be isolated from the rest of the acids and kept in a cabinet designated for corrosive chemicals. If a large chemical bottle is broken, evacuate the lab due to the fact that the vapors could potentially be dangerous. Always keep chemicals that are flammable or explosive away from ignition sources. Appropriate clothing and protective gear should be used for the handled chemical, especially caustic chemicals. Chemical disposal. There are 4 characteristics that define a waste to be hazardous: ignitability, corrosivity, reactivity, toxicity. Hazardous chemicals should never be poured down the drain or put in the trash. Hazardous chemicals should be disposed of according to state and local requirements. It is vital that waste containers are always labeled accurately. Waste bottles should never be filled up all the way. A few inches of space should be left at the top. Material Safety Data Sheet, or MSDS. It is vital to know as much as one can about the chemicals they are working with. The Material Safety Data Sheet, MSDS, is extremely helpful in this aspect, as it lists important information about the chemical, such as its properties, hazards, and safety precautions. Everyone in the lab should be aware of where these sheets are stored. These sheets include 16 sections, which include things such as composition, stability and reactivity, handling and storage, first aid, firefighting measures, and exposure hazard. Handling of biology specimens and waste. It is critical to follow all safety procedures and take proper care of biology specimens, preserved animals, fish, etc., to ensure they are used properly and are not degraded. Then they must be properly disposed. Preparation. Preserved specimens can be ordered through laboratory supply companies. Students should be informed of all safety guidelines before handling specimens. Gloves, aprons, and eye protection should be worn by all. Storage. Specimens can be stored at room temperature in the packaging they were shipped in. Some companies will ship specimens in pails while others ship in bags with a tissue fixative and preservation fluid. If all specimens in the container are not used, then the container can be closed up and put back in a storage closet for future use. Use. Dissection of specimens should be conducted with the use of goggles, aprons, and gloves, and be conducted in well-ventilated areas. Students should be reminded of all procedures relating to safety hazards before working with specimens. Disposal. Specimens ordered through laboratory supply companies can, in most cases, be disposed of in a school's regular waste. Most specimens do not represent a biohazard. It is recommended to always double-check with local authority and double-bag the specimens. Most preservation fluids can also be drained into the sewer system. If a specimen cannot be disposed of in a regular bag, then there are companies that can be contacted for removal. All specimens come with Material Safety Data Sheets, MSDS, which will inform users of unique disposal requirements. Dissection equipment. There is a variety of equipment used to dissect specimens in a biological lab. The equipment can come pre-packaged as a kit for easy use. Below is an example of an accessible kit that is used in dissection labs. It includes dissecting scissors, forceps, dissecting pins, a pipette, a dissecting needle, a scalpel, and a probe, among other things.